Research is the cornerstone of innovation in clinical practice, driving improvements in patient outcomes and shaping the future of medicine. Welcome to the Global Rhinology Network Topical Research Symposium. I am Ashok Rokade from Winchester, UK, and it is my privilege to welcome you as a host. Joining me as co-hosts are Louise Melia from Bath, UK, and Professor Thibault Van Zele from Ghent, Belgium. This symposium showcases fascinating topics across 4 key modules: General Rhinology, COVID session, and Research in Chronic Rhinosinusitis. A special highlight is the PhD session featuring research conducted by enthusiastic young rhinologists who represent the future of our field. This symposium brings together leading global minds in rhinology who will highlight latest developments with a focus on translating research into impactful patient care. The Global Rhinology Network is dedicated to advancing knowledge innovation. Thank you for being part of this exciting journey. Thanks to Medtronic and Karl Storz for their generous support in making this symposium possible. Welcome to GRN-Topical Research Symposium.
Noam, we're really Professor Noam Cohen. He is a Ralph Butler Endowed Professor from University of Pennsylvania. He's also a Director of Rhinology Research in the Institute, and he's also Chairman of a Resident Research Committee. So heavy involvement in research. His interest is in mucosal innate immunity and generally in chronic rhinosinusitis. Now we always have a strong support from Penn department. Professor Jim Palmer is our founding scientific committee member. He's always very popular on our live surgical workshops, and that's why it's another privilege to have you presenting the research side of the department. So over to you, Noam.
Again, thanks for the invitation. And what I'm going to talk to you about is some of the work that we've been doing over the last two and a half years. While the majority of the rhinology division here at Penn has been focusing on chronic rhinosinusitis, during the pandemic, we took a little bit of a detour into the world of virology, and I'm going to follow up on what Laura was talking about with regard to SARS-CoV-2. So as we know, the cells in the nose get infected before the cells in the lung, and that's evident early on in the pandemic with the high rate of olfactory disturbance. One of the things that was somewhat head-scratching to all of us is the paucity of other rhinologic symptoms, right? Most of these patients early on during COVID were not presenting with nasal obstruction or facial pressure, the classic rhinologic symptoms. It was anosmia, cough, fever, exhaustion. Less than 5% of individuals actually had rhinologic manifestations. And so this was actually one of the components that was incredibly scary about the SARS-CoV-2 infection, is that so many people were walking around infected but completely asymptomatic. And so one of the positions that we took is understanding what's happening in the nose before the virus actually gets into the lung is key in preventing the spread of this disease, even now in vaccinated individuals. The vaccines are preventing the severe respiratory compromise, but people are still walking around positive and still able to spread the virus in their nasal secretion. So understanding what's going on in the nose is going to be critical, not just for the vaccinated individuals now, but for any future coronavirus outbreaks. And I should mention that in the last 20 years, we've had 3 of these outbreaks already, and the next one is probably 5 to 7 years down the road. So we should get used to this, and we should learn as much as we can. And the key is what's happening in the nose. So in this schematic, I've kind of just gone very simply about what happens, how the virus gets into the nose. So it uses its spike protein to attach to a surface receptor. Basically, these spike proteins define all the coronaviruses. Each coronavirus binds to a different membrane receptor. In the instance of SARS-CoV-2, another common cold virus called NL63, they bind to ACE2. There's a proteolytic cleavage. The virus gets into the cell either through direct membrane fusion or through the endosomal route. It releases the RNA. The RNA then undergoes replication in something called double membrane vesicles. These are little outblebbings from the endoplasmic reticulum. The viral proteins are then transcribed and new viruses are assembled in the ER trans-Golgi and Golgi, where they form basically vesicles and then follow classic exocytotic pathways to be secreted into the respiratory secretions, where they either infect the neighboring cell, or they get aspirated into the lung to cause pneumonia and ARDS, or they get sneezed out to infect the neighboring person. The model that we've been using, and we've talked a lot about in so far today, the model that we use in our lab, is the air-liquid interface culture. We work with dissociated human nasal epithelial cells. We put them in what's called a transwell, grow them for a couple days. They form a monolayer. We can aspirate the media from the apical surface, and when these cells are grown in this air-liquid interface over the next 2 to 3 weeks, it forces them to differentiate into a ciliated epithelium. And this is what it looks like on bright field microscopy. You can see the beating cilia, scanning EM, and here's a cross section through one of those cultures where you can see that plastic pore support. Here are the pores, a pseudostratified epithelium, and a brush border. So early on in the pandemic, when everything was getting shut down, unless you were doing essential research, we decided not to sit on the sidelines, but to try to get in the game. And we reached out to Susan Weiss, who is a virologist at Penn and has been studying coronaviruses for the last 40 years. As a matter of fact, many of the proteases that a lot of the drugs that are in clinical practice right now against COVID, Susan was the one who identified many of these proteases back in the 80s and 90s. So Susan has been studying these viruses for about 40 years. I sent her a quick email saying, I've got human nasal epithelial cells. Would you be interested in trying to infect them? And she said, bring them over. So we ran 3 protocols initially. We took our human air-liquid interface cultures, infected them with 5 MOI of virus, let the virus sit on the apical surface for 1 hour, came in, aspirated it off, washed very well, and then let the infection pursue for 48 hours. We then collected the secretions on the top of the cell and performed plaque-forming units to determine whether there was any viable virus that was being shed off the secretions. We also collected the basolateral media to see if any virus actually made it through the cells and got into the media. And then finally, we fixed the cells and we stained for 2 different epitopes of the of SARS-CoV-2. One was for double-stranded RNA, and one was for the SARS-CoV-2 nucleocapsid antibody. So we had 2 different antibodies that we were using to validate which cells were infected. Another protocol that we tried was just putting the virus on for 48 hours and then doing the same thing. And the third one was to see if we could infect the cells through the basolateral media. I can tell you that this experiment completely failed. If you put as much virus as you want in here, barely any cells get infected. This experiment looked like a bomb went off with all the cells getting infected. And so we basically settled on this protocol. Put the virus on for 1 hour. This is your classic virology type of infection, wash it off and study the cells. And so this is what we got. Here's a mock infection. Give me 1 second while I shrink down my chat box. Okay, mock infection, the blue are the nuclei. Here in red is the nucleocapsid, and in green is an antibody to double-stranded RNA. So you can see nice co-localization of the double-stranded RNA and the viral nucleocapsid protein, and this is under higher power. Remember, the double-stranded RNA is staining those double membrane vesicles, and that's why we get these punctate patterns, whereas the nucleocapsid really is lighting up the entire cytoplasm. The other thing that you'll notice is that we see clusters of infected cells, and this is after 48 hours. The vast majority of cells are non-infected. We just get a couple cells that are infected. And if we counterstain for type 4 tubulin, which is going to light up the motile cilia, you can see that these clusters of positive cells are predominantly in ciliated cells. Every once in a while, we would see a discrete non-ciliated cell, but in general, over 98% of the time, the cells that were getting infected were ciliated cells. So as we were developing this platform for the SARS-CoV-2 infection, we started noticing some interesting things. One was that we saw variability in how many cells were getting infected after 2 days, depending on the individual that we grew the cells from. So this is donor 3019, donor 3575, and 4071, and you can see variable amounts of infection. And that's quantified over here, where we just literally counted number of DAPI nuclei and number of positive cells. So in 3019, about 25% of the cells were infected at 48 hours, and 3575, less than 4%. And in 4071, it was close to about 10%. So a variability in infection. And this pattern kept repeating, that certain donors got very well infected and other donors barely got infected at all. And we were scratching our heads as to what is going on with this variability when a paper came out. This was early on in the pandemic in New England Journal of Medicine, and this was the first GWAS study looking at COVID-19 severity. And the way that they defined severity was not only COVID-19 positive, so not just PCR positive, but some form of respiratory compromise, whether that was necessity for nasal cannula, intubation, ECMO. So there had to be some component of lung disruption for them to be severe COVID. They looked at about 2,000 patients and 1,500 controls, a fairly high resolution GWAS with about 8.5 million SNPs. And this is the results of that study. So they found two spots in the human genome, one on chromosome 3. So I'm sorry, so this is a Manhattan plot. Each one of these dots is those 8.5 million polymorphisms. To reach statistical significance at GWAS, you need a p-value of about 10 to the minus 8. So that's this orange dotted line. Anything above that is significant at the level of a GWAS, and they found two sites, one on chromosome 3 that associated with these 6 genes, and one site on chromosome 9 that just barely made significance that had to do with blood type. So blood type really didn't mean anything to me. I'm not a hematologist. It's too sophisticated for me. I was interested more in what was going on with this 6 genes. The only one that I recognized was CXCR6. It's a chemokine for macrophage chemotaxis. But the other 6, I had never really heard about that much. And so we ran those 6 genes against our human sinonasal single-cell RNA-seq database. And so what you have here on the y-axis is all the different cells that live in the human sinonasal cavity. Okay, so you got your dendritic cells, my favorite cell, the solitary chemosensory cell, your blood cells, your mast cells, monocytes, and then these are the epithelial cells. And against the x-axis are those 6 genes on chromosome 3 that associated with COVID-19 severity. And as you can see, we got a hit with one of those genes, LZ-TFL1, highly expressed and restricted really just to ciliated cells. Never heard of LZ-TFL1 before and I didn't know what it did, but it was worthwhile based on this data to buy an antibody for it. And so we stained for LZ-TFL1. And so here on the left is a biopsy from the inferior turbinate. You can see the basement membrane here, the pseudostratified epithelium. In green are the motile cilia, and in magenta is the antibody to LZ-TFL1. And this is the exact same staining pattern in our air-liquid interface culture. So you can see the little tufts of motile cilia all lighting up with LZ-TFL1. And when you look here at the epithelium, you'll see some black voids, and these are the goblet cells that inhabit the epithelium. So really, just like in our single-cell RNA-seq, the LZ-TFL1 was highly restricted and expressed in the ciliated cells. If we looked at the expression of this COVID-associated LZ-TFL1 in the airway, from the nose down to the bronchus, we can see differential expression. A lot of expression in the nose, although different between one biopsy and another, hardly any expression in the lower airway. So if this gene is really playing a role in COVID-19 severity, it's a little bit paradoxical because the way that they defined COVID-19 severity was respiratory compromise, yet LZ-TFL1 really isn't expressed in the lower airway. And so the argument that I'm making is the expression in the nose is really regulating what is going to ensue once the infection takes hold in the nose and allowing it to progress into the lungs. So as we were doing these studies, a bunch of other GWAS studies came up, and this is from the COVID-19 Genetics Initiative, and you can see these are 52 different GWAS studies, nearly 36,000 COVID-19 cases, 1.6 million controls. This is the Million Veteran Program. This is our Penn Medicine Biobank. You can see the UK Biobank is in there. So a whole bunch of different GWAS studies across the globe. Looking again at chromosome 3, focusing in on that area of LZ-TFL1. And now when you get up to these types of numbers, the significance is way above 10 to the minus 8. It's approaching 10 to the minus 23rd. So a significant genetic hit in the promoter region and in the 5' region of LZ-TFL1 in the context of COVID-19 severity. Now, as this data was coming out, there was another paper that came out in Nature correlating this part of chromosome 3 to the Neanderthal gene. And we should all be very interested in the Neanderthal gene. That's what the Nobel Prize was awarded for this year. And in orange are the polymorphisms that correlate to this Neanderthal gene, and in gray are non-Neanderthal. And you can see that the vast majority of the polymorphisms that were correlating with COVID-19 severity came from the Neanderthal haplotype. Now, why is this important and why am I bringing this up? If we look at the global distribution of this Neanderthal haplotype, we see that it is highly enriched in the Indian subcontinent. And as the pandemic was spreading across the globe, at least here in the United States, we were very concerned as to what the virus was going to do in Africa. Without the advanced healthcare that we have in Europe and in the United States and in Canada, we were really thinking that COVID-19 was going to decimate the African continent. And while the infection rate was very high in Africa, the mortality rate actually was exceptionally low. And one of the arguments, once that this data was coming to fruition, was that, well, people in Africa, it's a much younger continent. The older people tend to die off, or that their immune system is so jacked up because they see so many other infectious diseases. But here is a genetic argument saying that there is none of this COVID-19 or Neanderthal haplotype sequence found in the African continent. If you look at where it's most enriched, it's really in Bangladesh and in India. And if we think about what COVID did in India, and now again, it was the Delta variant, but the mortality rate was substantially higher than expected. And the study that I'm referencing here came out of London, and they basically said that people in London of Bangladeshi descent had twice as likely risk of dying from COVID than any other race. And it may have to do because they were 50% of them were carriers for this COVID-19 chromosome 3 risk allele. Okay, so that's the genetic component. What does this LZTFL1 do? So at the beginning of the pandemic, there were about 4 papers published on LZTFL1 function, and it looks like it plays a role in regulating exocytosis in ciliated cells. So when you want to secrete something in a ciliated cell, it comes out of the Golgi as a vesicle, it gets transported through intracellular transport. And then in most cells that are non-ciliated, that vesicle will just fuse with the membrane and release its contents into the apical surface, and you secrete. In ciliated cells, there's an option to transport that vesicle from intracellular transport to intraflagellar transport. And that's regulated by a large complex of the Bardet-Biedl syndrome complex that basically shuttles the cargo, these vesicles coming off the Golgi, through the ciliary necklace into the intraflagellar transport. It is argued that LZTFL1 negatively regulates that transport of post-Golgi vesicles from the cytoplasm into the cilia. And so that is at least what was thinking. And so at the beginning of the pandemic, when everyone was looking at the COVID-19 severity, people were focusing on ACE2 expression or the protease activation and how efficiently the virus was actually getting into the cell. But with this GWAS data and the implications in LZTFL1, maybe the real key to COVID-19 severity is not how efficiently the virus gets in, but more importantly, how efficiently the virus is getting out. And if you're very efficient at getting out, you're just putting more virus out, which then increases the probability of aspiration into the lung or spreading. And so to ask that question, could LZTFL1 be playing a role in post-Golgi exocytosis of nascent virions? We did the following experiment. We went into our biobank, and we've got about 1,000 samples frozen down of primary cells that we have genetically characterized, and we genotyped them for this chromosome 3 COVID-19 risk allele. And so we went after 4 different polymorphisms, and I should say that there are over 60 SNPs in this region, and they all tend to segregate together. So we genotyped 4 of the 50 COVID-19 associated polymorphisms, and we grew up patients that were homozygote high risk, homozygote heterozygote, and 1 homozygote low risk. And I should say that in the West Philadelphia population, we only found about 1% of the patients were homozygote high risk for that. And early on, I had a total of 9 patients that had the right genotype, but I had already blown through 8 out of those 9 donors. So I only had one donor that was homozygote high risk. Nonetheless, We grew up these cells from these 3 different genotypes. We ran our infection, and we looked early, medium, and late in the infection, collecting the mucus and doing PFUs, and then fixing and looking for the amount of cells that got infected. And this is what we got. So at 24 hours post-infection, again, in red are the infected cells. In the homozygote low-risk culture, we see 2 cells, 2 discrete cells infected. In the heterozygote culture, we see a couple clusters of about 4 to 5 cells. And in the homozygote high risk, we get a large cluster of about 50 to 60 positive cells. Here we've quantified the amount of cells infected. And here is looking at the amount of virus that was being shed into the airway surface liquid. And you can see nearly a log difference in this culture compared to this culture. Again, not statistically significant, but a trend that the homozygote high risk was putting out 10 times the amount of virus as the homozygote low risk. That's at 24 hours. At 48 hours, the homozygote low risk now looks like the heterozygote with bunches of clusters of cells of about 4 to 10 cells. The heterozygote now looks like the homozygote high risk from 24 hours prior with a lot of big clusters of infected cells. And in the homozygote high risk, the vast majority of the cells are infected. Again, quantifying it, 5% of the cells infected here, almost 20% of the cells infected up here. And now when we look at the amount of virus being shed into the airway surface liquid, we have statistically significant increase of over a log, over 10 times the amount of virus being shed here than there. And at 72 hours, we're counterstaining now for EBCAM to look at the integrity of the barrier, right? So we're looking for healthy looking cells. And at 72 hours in a homozygote low risk, you can see a large cluster of infected cells, but the cells are pretty much intact. They're having this hexagonal or octagonal shape. And overall, the barrier looks completely normal with the exception of a couple infected cells. In the heterozygote, we start seeing some balling. So the cells are starting to die here, but overall, at 72 hours in the heterozygote, the barrier is intact. And when we look at the homozygote high risk, it looks like a hand grenade went off. A lot of these cells are undergoing apoptosis. The barrier is falling apart. Basically, very, very different picture in a homozygote high risk compared to a homozygote low risk. So going back to our working hypothesis that LZTFL1 is negatively regulating how much virus is making it out of the cell, we would think that there would be an inverse relationship. More LZTFL1 expression, less viral exocytosis. And so luckily the antibody that we bought was actually very good by Western. Here you can see the full gel, not just a cutout, very, very clean blot for LZTFL1. Here it is in the low risk, heterozygote, high risk. In some patients, we see a doublet, and some we see a singlet. Don't necessarily know what the doublet is. But if we then counterstain for GAPDH and we do densitometry for looking for protein expression, you can make the argument that there's less LZTFL1 in the high risk compared to the low risk individual. And here's a whole bunch of other samples from the heterozygotes and the low risks. And if we basically do densitometry of this band over that band and we graph it out, we can see that in the homozygote high risk and even in the heterozygote, there's a trend of decreased expression in the high risk or the heterozygote individuals. Now, the other thing I should say is that we also looked at some cell lines, and lucky for us, A549, which is an airway cell line, do not express any endogenous LZTFL1. So from an experimental standpoint, this is very good because we can then transfect in LZTFL1 with ACE2 and ask, what does that do to the infection? And so here you can see we're growing our ACE2, our A549s either without or with ACE2, without or with LZTFL1, grown in submersion or grown in air-liquid interface. As the cells differentiate to air, they decrease their expression of ACE2, but there's still ACE2 there, and we get nice expression of LZTFL1. So here again in red are the infected cells, and you can make the argument that when you transfect in LZTFL1, there's more infected cells than without, but relatively good infection. But when we look at the amount of virus that is being shed either into the media or into the apical surface, in each one of these conditions, whether it's done in submersion or whether it's done in air-liquid interface at 24 or at 48 hours, we see a decrease in the amount of virus that's making it out of the cell into the media, going along with our hypothesis that LZTFL1 is negatively regulating virion exocytosis. And so that's the model that we're currently working at. The problem is if we would think that LZTFL1 is really playing a role in vesicle exocytosis, we would expect enrichment really up here in the apical side. And as you can see by the immunostaining, it's really lighting up the entire cytoplasm. And so is there something else that is membrane associated that has to do with viral replication or viral exocytosis that LZTFL1 could be doing that doesn't really involve the basal body or the cilia? And I mentioned to you that the virus, the RNA, is replicated in these double membrane vesicles, right? These are little membrane pouchings from the endoplasmic reticulum. And using our double-stranded RNA, we can actually quantify the amount of these double membrane vesicles that are basically RNA factories for the replication of the RNA. And if we look at the low risk, again, we only get one cell infected compared to the high risk, but if we count up the amount of puncta per nuclei, it looks like there's more double membrane vesicles in the LZTFL1 high-risk individual compared to the low risk. So maybe LZTFL1, because it's ubiquitously expressed, is somehow or another helping to stabilize these double membrane vesicles and make it more efficient for the virus to replicate its RNA. So I'm going to conclude with basically these are preliminary findings. We're trying to increase our numbers. We're running into issues with getting homozygote high-risk individuals to replicate some of these studies. But what I've shown you is that there's significant GWAS data out there suggesting that LZTFL1 somehow or another is associated with respiratory compromise associated with COVID-19. LZTFL1 is restricted to expression in ciliated cells. It appears that there's an inverse relationship between that there's a decreasing gradient of expression of LZTFL1 as we go into the distal airways, and that there's an inverse relationship between LZTFL1 and SARS-CoV-2 in vitro infection. And we also think that in addition to playing a role in regulating the exocytosis, LZTFL1 may be doing something with regard to stability to these double membrane vesicles. I'm going to finish up with one last picture slide talking about the variants. And so we can also use these air-liquid interface cultures to study the variants of concern. And so what I'm showing you here are cells from the same individual infected with the original Wuhan strain, the Delta and Omicron at 24 hours or 4 days later. This is what we see with the wild type. This is Delta, and this is Omicron. Although they are, the same virus, clearly a very, very different phenotype in what these different variants are doing in the nasal epithelium. And so this is another area that we're focusing in now on trying to understand what is Delta doing to form these big syncytia, whereas Omicron is infecting a lot more cells, but not forming any syncytia. And so with that, I just want to mention my collaborators. None of this would be possible without the collaboration with Susan Weiss, who is the vice chair of microbiology and runs the BSL-3 facility over at Penn. There are 9 people in her lab currently working on different projects with all the different coronaviruses, including MERS in the nasal epithelium, my genetics collaborator over at Manel, and the people in my division who have been making this all possible. And so with that, I will end, I will stop sharing, and I will take any questions. Can't hear.
Hello, Thibault. Sorry, you're muted.
Can I ask a question, please?
Sure.
Do you think that going forward, this is information that we could use to identify, a cohort of people that could be vaccinated, rather than the entire population?
Yeah. So if we had gotten our act together faster, and as the vaccines were rolling out, but they were limited, you can make an argument that, again, I don't want to get into the ethics of this, but the people that are high risk, the people who are carriers for this chromosome 3 locus, probably should have been moved up to the front of the list with regard to vaccination. That the people that are homozygote low risk, these are the ones that are completely asymptomatic, where it really is, you don't even know you're infected. The heterozygotes are probably moderately symptomatic, but it's really the homozygote high risks that are at much greater risk for respiratory compromise and ARDS. What we're trying to do is go back now retrospectively and genotype all the patients that were in the MICU and ICU and try to make that statement, really corroborate that with data. But based on what we've done and what some other people have done with regard to this locus, this is really one of the keys with regard to how efficiently it's making it through the nose. So yeah, I think that, from a epidemiologic or from a public health standpoint, something like this could be implemented. Now, if you think about it from an ethics standpoint, it's a Pandora's box, because then what else are you testing for? But when the vaccines were just getting rolled out and there were limited supply, this probably would have made sense as a way to identify who should really get to the front of the list for vaccination.
Thank you.
Just one question. Hi, and that is, what role does the age play here? Because in the beginning it was all elderly people and things like that. So how do you see that one?
So the way that I think about this is this is pretty much all in the nose. The way that it gets into the lungs is through aspiration. As we get older, we microaspirate more, right? The larynx, the sensation of the larynx. I mean, we see this all the time in the clinic, right? We have these older people that are microaspirating. The other thing that we see as we get older is more vasomotor rhinitis. And so I think that the stuff that we deal with and we've dealt with for the last 100 years as rhinologists, which is the common things, microaspiration and vasomotor rhinitis, which both get worse as we get older, helps the virus work its way into the lungs. The more aqueous the secretions, the more volume there is, it's being shed into the mucus. You're either going to swallow it or you're going to cough it out. But if you aspirate it, once it gets into the lungs, the work that we've done with the pulmonary people and the alveolar cells, this thing wreaks havoc. Like I showed you in the 72 hours, the homozygote low risk, the cells were completely intact. Once that the alveolar cells get this virus, they fall apart. Within 24 hours, they're undergoing apoptosis and necrosis. So the lung cells are exquisitely more sensitive to the virus than the epithelial cells up in the nose and even in the trachea.
Thanks.
So I think it all has to do with the kinetics. The other thing is that if you get the virus and it takes a while for it to mount and start shedding into your secretions, it gives your body a chance to mount an immune response. In the homozygote high risk, they're shedding so much virus so much faster that the virus is ahead of your body being able to actually mount an immune response. So it's all about the timing and that initial infection. How fast is it moving through the nose? That if it's slow, your body will get caught up to it, kind of like while we were vaccinating. Whereas if it's too fast, it's going to end up in the lungs before there's an adaptive immune response.
Okay.
Okay, that's interesting. Noam, does this research in any way help us to build any model for future pandemic management?
Well, one of the things that we're focusing on is I never really understood why we were doing intramuscular vaccinations, right? I mean, we're getting basically our antibodies in the blood. What we really need to be focusing on is an IgE, a secretory antibody in the nose. The nasal secretions are going to be key for all of these respiratory viruses. The reason why we went with the mRNA going into the muscle is because all of the big pharmaceutical companies are built for injection. If we, and we're actually, the mRNA technology is sitting right across the street. That's in Drew Weissman's lab. So we're trying to see if we can actually take the mRNA encoding different epitopes, put it into our nasal cells to see if we can actually get production of either spike protein there to induce IgA, or more importantly, could we get the epithelial cells to make antibodies that are designed against COVID? So the platform, I think that people are really starting to focus on now that a lot of these respiratory viruses, while we as rhinologists knew the nose is key, big pharma is now starting to appreciate that we have to focus in on the nose as really the critical part to do this. So did that answer your question?
Yes, in a way. And does that make the vaccine slightly more safer than compared to the intramuscular?
Well, the vaccines are very safe. I don't, what do you mean by
that's the baseline? Yeah.
I mean, at baseline, they're incredibly safe. Could we, the question is could we somehow or another formulate it for an intranasal type of vaccination that would get the immune cells in the nose to start making secretory antibody? And that way, if we can neutralize the antibody in the respiratory secretions, we don't have to worry about it getting anywhere else.
Okay, thank you.
So could we use these ALIs as a platform for that? Well, maybe in a year or two, I'll be able to let you know.
Okay, thank you.